Our first session of the afternoon after that awesome song is Inside HSG and Current Trials, and will be led by Dr. Victor Sung from the University of Alabama. Over there, we see you, Dr. Sung. Please welcome Dr. Sung. You gonna stay down there? You're coming up. I know you like to wander around.
Yeah.
I know your style. I've been watching him for 2 days, you know what I mean?
I'll just need the clicker.
All right. You're gonna stay down there.
Yeah.
All right, take it away, Dr. Sung.
All right, good afternoon, everybody. I'm going to try to keep you awake after lunch. I always say I can't do stand behind a podium thing. I have to rove around. I'm going to attempt to go through a research blitz in 30 minutes. I don't know how I'm going to do that, but it's helpful that Martha has set up a lot of things, and you've had some research talks already to set the stage, but I'm going to try to provide a framework and go through some of the main trials, and then also Dr. Hernandez is going to come after me and follow up more with these things. So this is how I'm going to lay it out. We're going to talk about the HSG trials. I'm going to take you through some things that you need to know and understand to understand all that's going on now and in the future in HD. I might stand up here so you can see me better. But, and then I'm going to try to go through the data and explain it in a patient and family understandable way in 5 different trials in 30 minutes. All right. So for the HSG trials, the big ones that are going on now are really all based off of this My HD Story platform. I put the QR code up here big. All of you just take a picture of it, sign up, and we don't expect you to, I actually don't want you to do the surveys now. I want you to listen to me, but you click on it, go to the website, and all you have to do is register. The My HD Story platform is this, you register and then studies will come to you and you go in your own time and fill out surveys. These filling out of surveys is what we call patient-oriented research, and we're wanting you to tell us your story, right? And it's a way to get involved in research from your own home. For your family members who maybe don't want to come into clinic, I know neurologists like us are really scary, even when we're not roving around on a podium. So this is a way to do it from the comfort of your own home, okay? The 2 open studies are Project Aware 2.0 and LEAD HD. They're assessing these patient-reported outcome measures. So go to the website, sign up today, all right? Hopefully, I saw lots of you with your cameras up. That is the big thing. All right, so let's lay the framework for talking about research. Dr. Nance talked about HDISS. I'm going to come back to that a little bit. But I think there are things that you have to understand so that when you see press releases, when you see things in the news about, oh, they meet these endpoints and blah, 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 all this jargon, what does it all really mean? So to lay the framework, I want to take you through some of these things and try to explain it, right? So most therapies to date have really focused on, and we're talking about disease modifying, right? Trying to slow the progression of the disease. The traditional model has really been focused on Huntingtin lowering, meaning if you look up in the upper left, you have the DNA, which is this double-stranded thing, and then you make this mRNA message, and then it produces this protein, this Huntingtin protein, and the theory is that this Huntingtin protein causes damage and kills brain cells, and that causes the problems that we get in Huntington's disease, right? So then if we can make less of that protein, that's called Huntingtin lowering, and if you reduce the amount of the bad stuff that's causing damage to cells, that's how we fix this disease, right? That's been our traditional understanding, and that's what a lot of the therapies are going after. And not that that's not going to yield things, but there's a few things that we've known about for a while that have always been in the back of our mind. We didn't have a good explanation for it, but we didn't have a better explanation for it, so we just don't talk about it. And some of that is you guys know how it is, right? It's 50-50, right? You have one mutated allele and you have a normal allele, right? So it's 50-50 if you pass it on to your child. If you pass them the mutated allele, they're going to get it. If you pass them the normal, they're not. But we all know that this stuff happens, right? Like, we have all of these stories. You have two patients who meet in support group and fall in love, and then you have two parents that both have Huntington's disease, and then it's possible that they could have a child with two bad copies, right? Two mutated copies if both parents have Huntington's disease. We know this happens. It's rare, but it happens. I'm particularly proud of, even though I practice in Alabama and all the stigma about Alabama, I've never had this happen in my practice. I'm proud that has not happened, but anyway, well, we know this happens. Well, the people that have two copies of the mutated allele, well, what is that? Well, then they make twice as much of that bad protein. But interestingly, they don't have twice as bad Huntington's disease. So that's weird, right? So if this is the whole story, that the only source of the problem in Huntington's disease is this bad protein causing toxic damage to kill brain cells, and that cannot explain that people that have twice as much of the bad protein are not twice as bad with their Huntington's disease, right? So there must be more to the story, and we've known about this, but we don't talk about it because we don't have a better answer until recently. So you'll start to hear some things in the literature or in public things where they talk about this thing called somatic instability or somatic expansion. What the heck does that mean? When you went through testing, probably the doctor who did your testing said what I tell all my patients, which is, if you test when you're 5 and you test now, or you test 10 years from now, your test should be the same, right? Because I have some patients who are like, okay, I've tested positive for HD. When do I need to retest it? I'm like, you don't. You've tested, it's positive, it's not going to change. And that is true, asterisk. So, not that we've been lying to you all these years, but that is true in blood cells where we measure your Huntington's repeat length, right, when you get a blood test and get your gene test. That is true in blood cells, it stays the same. What we know now, though, is that in brain cells, particularly the brain cells that are involved in Huntington's disease, at the end of life, your 42 CAG repeats in those brain cells that are rapidly dying off. We know this from autopsy studies, that they're super high, like 400, 500 CAG repeats. So what happened, right? In the rest of the body, this 42 CAG repeats that you might have is in every single cell of your body, but in most of the body, it stays the same. Well, what happens in brain cells that it goes haywire like that, right? So anybody have kids that play with zippers? And they played with the zipper and they messed it up and the zipper got off track and the whole thing's falling off and you're like, we're trying to leave. And your zipper's all messed up and you can't go out of the house like this, And of course it's like right when you're trying to go somewhere, that there's zippers like this and you're like, what is happening here, right? So if we go back and you have this double-stranded DNA message, right? So there's two parts of the DNA molecule. You can think about it like the two sides of a zipper, right? So to open up and make the message and make the protein, the DNA has to open itself up, make the message, and then zip itself back up again, okay? Well, the problem is when you have a lot of the same in a row, like the CAGs over and over again, the more you open and close like that, and open it, stamp it out, and close it, every once in a while you get this zip failure, right? So it's like the zipper getting stuck, and you get this bubble, and the zipper's off track, and your jacket's like this. Well, what the body does, they don't like that, right? So they go in, and with repair mechanisms, they fix it to try to straighten the zipper out again. And the way they do it is they insert a few more CAGs to straighten it out, right? Well, now what's happened? If you insert in a few more CAGs to let the DNA zip closed again, now your 42 is a 45, right? Forever. And then it happens again, and it becomes a 48 and a 55. And so that is what's happening, and that is controlled by certain other DNA repair proteins that tend to fix these zip failures by sticking in more CAGs, right? And guess what? We know that Huntington's patients have mutations, or frequently have mutations in those other proteins as well, that tend to get overzealous with sticking in more CAGs and making it do this, right? So what we know now then is that in addition to the toxic protein causing problems as one of the mechanisms for Huntington's disease, this CAG repeat getting super long can be a problem too. So it's probably both things contributing to what causes the underlying mechanism of the disease. Well, it makes it more complicated, but it gives all the scientists here another target to go after of something else to try to address this difficult disease, right? It gives us more ways up the mountain to try to solve this problem, okay? So that's somatic expansion or somatic instability. It's this runaway, you're 42 becoming 500 in a brain cell. That's another part of the problem, and that's something that we're trying to correct. Does it make sense? Okay. Another thing that you'll hear about when you read in like press releases, things like that, there's this thing called NFL. And obviously, we live in Cincinnati. We're not talking about the Bengals. In HG world, NFL stands for neurofilament light. So what does that mean? The neuron, I have a picture of a neuron, which is a brain cell on the right. The long part of it has filaments in it to hold it up. It's like a string, right? Do you see the long tail there? There's filaments in there, what's a filament? A filament is a string, right? You know, if you're any fishers here, like what do they call it? It's a monofilament line, right? So a filament is a string, so there's strings in there to make that long tail. It's like the scaffolding, or it's like the framing of your house, right? Do we get tornadoes in Cincinnati? Probably, right? So if you have a tornado come through Cincinnati, what happens? You see all this wood debris everywhere if it's blown through your neighborhood, right? Because what's happened? It's knocked the house down, and all the framing, the wood stuff, is all spewed out in the street, okay? So that's what's happening in Huntington's disease. Think about Huntington's disease as your tornado ripping through your neighborhood of your brain, right? It kills a brain cell, and it spits out the framing, which is this neurofilament, right? So if neurofilament, or NFL, if there are large quantities of it floating in your bloodstream or in your spinal fluid where we can measure it, that's bad. Why is that bad? It's a sign that brain cells have been killed, right? A tornado has come through. So it's not specific for Huntington's disease, though. Like, it's interesting that I have the NFL shield up here, right? What happens if you have an NFL concussion, right? Guess what? If we test NFL players after they have a major concussion, what will we find? Their NFL levels will be up. Why? Because you killed a bunch of brain cells when you had this head-to-head collision. Does that make sense? So anything that is killing brain cells will cause NFL to go up. So in general, when we look at studies, research studies, we don't want to see big spikes in NFL. Does that make sense? Because what if the drug that we're testing is doing the right thing, it's lowering mutant huntingtin, or maybe it's fixing the somatic expansion, but you see this big spike in NFL, it could mean that the drug itself is causing damage to brain cells, and that's not good. Does that make sense? So in general, when you're looking at research results, the thing that you should look at for NFL is that big spikes or it going up is bad, right? We don't want NFL to be going up. We want NFL to be going down, okay? If we do nothing in Huntington's disease, in general, we don't know this perfectly, but in general, we know that Huntington's patients, their NFL levels will do what over time? They continue to go up, because why? The disease itself is killing more brain cells over time, okay? So that's why this could be a marker of the disease itself and things like that. So, but just think about that. NFL going up, bad. Mutant huntingtin protein going up, that's probably also bad. So we want those things to be low, all right? Then Dr. Nance also introduced this HD integrated staging system. She did a nice explanation of it. I'm not going to go through that again. But what I want you to see is like the whole point of the HDISS, right, is not that we don't care about the symptomatic patients or the more advanced symptomatic patients anymore. It's that, think about it from this standpoint. Like these first drugs out of the gate that are trying to be disease modifying and to fix this problem that's Huntington's disease. What's the likelihood that the first drug across the line is going to be perfect and completely stops the disease and has no side effects? That's like zero, right? Like it's not going to happen. Most likely, the first ones across the line are going to slow the progression of the disease and not stop it completely. And maybe a combination of them or better ones over time will be able to stop the disease completely, right? But I know what you're thinking. It's probably what I'm thinking, what I tell my patients. If we can slow the progression of the disease and not stop it completely, I'll still take that, right? What if we can slow the progression of the disease and instead of death at 55, you get death at 65? What if instead of you have to retire from work at 45, you can retire from work at 65, right? That's still a huge win, right? So that's what we're hoping for. And if we're going to achieve that, slowing the progression of the disease, shouldn't we try to go earlier, right? Shouldn't we try to slow it before it's full blown, right? Think about your children. Think about your grandchildren. Wouldn't you want that? If your child has 42 CAG repeats and is 18 years old, right? And they're totally fine. They don't have any symptoms. But you had the opportunity to give them something that could slow the progression of the disease, and they have symptom onset at 55 instead of 45. I'll take that, and I'll want that for my 18-year-old child now. Does that make sense? So if we're going to go earlier, how are we going to do that? Are we going to start at like newborn screens or a 5-year-old? Like where's the line? That's hard. The only way we can do that is to define groups of patients that are early, right? And so the whole point of the HDISS is to put some boundaries on who is early, right? Because as Dr. Nance said, our old staging is really stage 1 is already symptomatic Huntington's disease. Well, we want to actually start before that, but then you have everything from 44 and you're going to start in 2 days with symptoms all the way down to newborn, right? And newborn screen. Does that make sense? So we have to put some bounds on that, and that's the whole point to have stage 0, stage 1, and stage 2, okay? So you have to understand these HDISS things because all the trials upcoming or ongoing are going to say, we're testing this study population, and they seem awfully mild, and it's like, I'm already past that point. Well, it's because they're trying to go earlier, and they're trying to achieve for your children and grandchildren what unfortunately maybe for you, if you're already in HDISS stage 3, the science hasn't gotten there that fast, right? I think about a lot like when in 1993 when the gene was first sequenced, right? I don't know if you remember, some of us remember what was in the news, right? In the news, in the press, it was, oh, we've sequenced this gene now. Now we know what causes this horrible disease, Huntington's disease. It's going to just be a matter of time. 5-10 years, we'll have this thing nipped in the bud and cured because we know what the target is now. And now, what, 30, 40 years later, we're still not there yet. So it's complicated and it's hard, but finally science is catching up to the things that we've wanted to do. When I took over as the Center of Excellence Director in Alabama, I say this all the time, my first HSG trial, my first Huntington's disease trial that I did was called CREST-E, right? Some of you may have done that trial. It's high-dose creatine powder for Huntington's disease, right? We did it because that's what there was to do. But in the back of my mind, I'm thinking, really? The cure for Huntington's disease has been on the shelf at GNC all this time and we didn't know? Probably not. But we did it because that's all we had to do. And then fast forward 10 years later, And now we're talking about manipulating DNA and doing all these fancy things, right? So the science is now finally catching up to all the things that we wanted to all along since 1993 when we sequenced this gene, okay? So, but the terminology, the lingo gets more and more complicated, and how do we do that? So this is showing DNA on the left, and then protein in blue, that's in green, and then protein in blue on the right. All these arrows are all the places in all the steps in the process that we can intervene, and guess what? We have companies doing things at all those steps in the process, which is cool, okay? I say bring it, you know? The more big companies that want to throw their hat in the ring in Huntington's disease, come on. There's room for all of us in the sandbox, you know what I mean? Okay, so I'm gonna dive through, let's see, how am I doing on time? Okay, 9 minutes left. So I'm gonna run through a bunch of slides on a bunch of specific programs and give you some of this data. So the Roche tominersen program, as you may remember, it was testing this antisense oligonucleotide, this string that can bind and block and try to lower Huntington that way that you have to inject in your spine. So it's a spinal tap injection. If you remember a few years ago, the study, Generation HD1, was stopped early. Why was it stopped? Patients were having, some patients were getting worse. So what do we see? Let's see, I don't know, do I have a pointer on here? I don't know. Okay, I'm gonna do it the old-fashioned way. So if you look here, the pink line is mutant Huntingtin protein, right? So do you see as we go up over time, these are different doses. What's happening with the pink line? It's going down, right? Do you see that? Everybody see that? The pink line's going down, that's good. That means mutant Huntingtin is going down. That's what we want to do. Well, why does the study get stopped early? What's bad? Do you see the orange line, the NFL, and the far right there? The highest dose group, what happened? NFL went way up, big spike in it. So we talked about, we want mutant Huntingtin to go down, we want NFL to go down, but this, it went up. But is the orange line going up in every single frame here? No, right? So the orange line is about flat in maybe but the pink line is going down in these middle two sections. Do you see this? Okay, thank you. Oh, okay. Which one? The
bottom green one?
Oh, yeah. Okay, got it. So these middle two here, the pink line is going down and the orange line doesn't spike, right? So what Roche said is like, well, what if we look at this and not do this high dose over here, but what if we go back and do lower doses over here? Maybe some lowering is good, but we don't see the spike, and maybe that will be good. And when they looked at those patients actually were doing better from the drug. So it was worth revisiting, and they've rebooted that with another trial, okay? So that's the why, and that's where that comes from. When they separated out all those patients, then it's not enough to prove it statistically, so they got to redo it in another study, but they're redoing it in another study looking primarily in these groups here in the middle. Does that make sense? Okay. What I'm going to take you through the rest of the time, the easiest way to do it, there's a million studies. I can't cover them all. Dr. Hernandez actually will run through more of the studies than I will. But I'm going to take you a little bit deeper into the big four, what I call the big four. The history-wise is we've never in the history of Huntington's disease had 4 consecutive press releases that were positive for disease-modifying therapy in Huntington's disease. So 2024 has been a good year, right? Because if, you know, as Dr. Kostic was saying earlier, she's had how many failed trials that she's had to do? And that's been our experience. It's like negative news again, you know, and this defeat. But we've had 4 good news. I mean, there's still work to do, but 4 good news in a row, like, we'll take it, right? After what we've all been through as a community, we'll take it. So let's go through these press releases in order. I'm going to break them down for you, help you to understand them a little bit better. So the first one was from Wave. This is a screenshot from the press release. Shows allele selective mutant Huntingtin reduction in phase 1b/2a select HD trial. Great. What does that mean? So what does Wave do? So their drug, instead of the CAG repeat itself, it targets a little stretch of DNA that's down from the CAG repeat itself, but connected to it. Does that make sense? So it targets that and knocks it down. Well, do you see in the illustration here, on the normal one, there's a green little strip there, and on the mutated one, there's a red one, right? So what they've developed now is anti-red, and then it will just knock this down and leave the normal copy alone, right? So that's what their drug is trying to do. Now, what does that mean, though? That means if they've developed anti-red, right, and what if you have green on your mutant allele? Well, anti-red's not going to work for you, right? What if you have red on both copies, the normal one and the mutated one? Well, also, anti-red is not going to work for you because it'll knock down both, and we're trying to just go after the mutated one, right? So that's true. Even if this works, it won't fix everybody, but guess what? If their drug works, they have anti-green in the cupboard that they're ready to pull out. They have anti-blue in the cupboard ready to pull out so that it will cover all the things no matter which one you have on down from your mutated expansion. Does that make sense? So we got to do the work in increments. No one thing is going to solve it for everybody, but this is still good that we're headed in the right direction. This is from their press release. What do we see here? We see that the blue mutant Huntingtin protein went down. That's good. Wild type, that's this over here, that's the normal copy. Normal copy was about the same, but didn't go down. So that means it's doing what they want to do. It's knocking down the mutant, leaving the normal copy alone, okay? Great, we will take it. What about NFL? So this is their NFL data. We see a spike here. So not in every patient, but some patients had some big spikes in NFL. I don't know, that's not great, but there's more work to do. It's a small number of patients. We've got to understand it a little bit better. A little bit of cause for concern maybe, but we'll have to see. This is a small study. We'll have to do more, see more. But this is still pretty good, right? They were able to knock down the mutant, but leave the normal alone, okay? All right. And this showed that this is the motor score. This is this stuff. You guys ever been in clinic and we make you do all this stuff? What's everybody's favorite? This one, right? That means that is getting better. You see? So the blue is the treated patients. So the treated patients got better on their speed at doing this stuff. That's good, right? So mutant Huntington's going down. Their ability to do some of these things is getting better. The only negative thing is the NFL is going up. We have to explain that more, but at least it's a start, okay? What's going on with WAVE is they've submitted all their stuff to FDA, let FDA look at it. They'll be ready to launch a big phase 3, does this work or does it not trial sometime in 25. So stay tuned for that. That's exciting, and the ASOs against SNP1 and SNP2, that's the anti-green, anti-blue that I was talking about. They've got that in the cupboard. They're ready to pull that out if this works, okay? All right, good news number 2, Unicure. That's the gene therapy. Gene therapy requires brain surgery and a direct injection into the brain, right? Super invasive. They announced positive top-line data. So let's break that down a little bit. It's brain surgery. Like shave your head, drill holes in your skull, put catheters in, pump stuff into your brain. That's crazy. It's like a 16-hour surgery, too. I get it. Some patients are like, yeah, brain surgery for Huntington's disease. Why don't you come back to me when you've proven that it's safe? I'm like, I get that. Some patients are like, my window for treating Huntington's disease and slowing the progression is small. And so you're saying, I can go to the operating room, you can put me to sleep, and I wake up and the cure's in my brain? Or worst case scenario, something bad happens in the surgery, I don't wake up. Well, I've got Huntington's disease, there might be worse things than that, right? And so they'll say, sign me up. And I get that. Like, every person's going to be different. But for some people, they may want to do this, right? So I have 7 of my patients that got treated, that got surgery. This is about 3 years ago now. And I'm still seeing them, and they're still doing stuff, right? Like, One of my patients, in particular, I just saw him like last week, and I saw on the chart that he had a birthday. And I was like, oh, you just had a birthday. Belated happy birthday. And he started crying. And I said, wait, what happened? And he said, I was like, I just said happy birthday. What did I do? Like, I stepped in something, you know? And he said, no, the birthday that I had is the same age that my mom was when she died of HD. And we have the same CAG repeat number. And I'm still working full time, and I just won a productivity award, like a sales award at work. At the same age, with the same CAG repeat number as that my mom was when she died of this disease. And he can't, I don't know how to explain it any other way than he got this treatment, you know? He's the same, if not better, than he was 3.5 years ago when we put him in this trial before his surgery. Like, that's not the way HD normally goes. I don't know. Like, yes, there's placebo effect, and we've got to work this out, but I don't know. It's interesting. And this is their data, okay? I like to tell stories, which is bad. It wastes all my time. But this is showing that without the treatment, patients get worse. We know that. That's HD, right? With the treatment, patients get a little less worse. That's this stuff. The CUHDRS, if you ever see it in trials, okay, that stands for composite UHDRS. What that's doing is combining your score on this stuff, this tapping stuff, and your score on the red, blue, green, blue, blue, green. Anybody done that? Yes? It's combining all of that together. That's why it's composite, right? So patients that got the drug did a little less bad on that, okay? And what did they see with NFL? So obviously, we drilled 4 holes in your skull and poked 4 things in your brain. After the surgery, we saw a big spike. But interestingly, 2 years out after the surgery, all the patients on NFL are lower than where they started 2 years prior. And we talked about what? Like NFL and HD should go what over time? It should go up, not down, right? So this is kind of interesting. We'll see. It's 26 patients. We don't know, right? But there's enough smoke there that we should see if there's fire there. Does that make sense? All right. So they're meeting with FDA this month. They're going to ask for accelerated approval. So we will see what they say. Fingers crossed. All right. Sorry, I'm over time. Is it okay if I keep going a little bit, or do I need? Okay. All right. So good news number 3. PTC reports positive data, FDA lifts partial clinical hold. I actually want to explain both of those things. Do you guys know why it was put on a clinical hold in the first place? Does anybody know? So what is PTC? PTC has a pill that you can take to try to slow the progression of Huntington's disease, right? Well, the FDA put it on a clinical hold for one reason and one reason only, and that's because PTC, when they wanted to launch in the United States, they said, we respect the HD community and your time and your effort to do these clinical trials, which is hard, right? Who in here, someone here has done a bunch of trials with Dr. Duker, right? It's hard. It's hard work, right? And so PTC said, we want to respect that, and if you got placebo in the first part of the study, we want to let you get the switch over when you're done with the research part and switch over and get the real drug for free until we get FDA approval. And the FDA said, no. You cannot promise to give patients real drug until you've proven that it's safe. We want to protect our American citizens from evil pharmaceutical companies like PTC or whatever. And so they said, no, that's why they put it on hold, because PTC wanted to offer what we call an open label extension to allow patients to have access to the drug for free that were in the trial. And the FDA said no. So, but now the drug is shown to be super safe, and so now they're going to lift it now. But that means that we did the whole study outside of the US, and none of us got to participate. Okay, that's fine. I got a meeting with the FDA next week. You know, we're going to knock some heads next week. All right, so So what does the PTC drug do? So what you'll see is these small molecule splicing modifiers. Small molecule, if you ever see that anywhere you're Googling something, Huntington's treatment, it says small molecule. Small molecule means you can take it by mouth, you can eat it. So that's good, right? Pill is a lot easier to take than brain surgery or spinal tap injection, okay? What they do is they stick a stop sign in so that the whole message doesn't get translated. It just interrupts it and it stops it there. Well, it's a pill that has to go everywhere to get in the brain. It's not going to completely stop us making the Huntington's signal everywhere in all cells in the brain, but it will knock it down some, which will take it, okay? So what did their data show? Their data showed that this is the mutant Huntington levels. So patients that got placebo or sugar pill, mutant Huntington went up. Well, that's Huntington's disease, right? Patients that got the pill, mutant Huntington went down. Good. That's what the drug is supposed to do, okay? And these were NFL levels, no spike. So mutant Huntington going down, no NFL going up, that's good. We'll take it, okay? This is just to show that they got better on some of these measures and stuff too, okay? And they're basically like minimal side effects. So they're gearing up. We expect a phase 3 trial to start in the US sometime next year. So stay tuned for this. This is exciting too, okay? When they do it, we don't know exactly who they want yet, but probably late stage 2, early stage 3. So we'll see. ISS stage. Good news number 4, Skyhawk. Okay, so this was their press release. What was this day? July 10th, okay? Like I said, it was a good summer. So they have another one of these pill splicing modifiers, right, where it sticks the stop sign in and messes with the production of the mutant Huntington protein. But interestingly, their drug puts a stop sign in at 2 places. One, in the mutant Huntington gene to produce mutant Huntington protein, and 2, in this other gene called PMS1, it interferes with the production of that protein too. Well, what's PMS1? Guess what? It's one of those zipper repair things. So possibly, Skyhawk can interrupt both mechanisms, make less Huntington, and maybe disrupt this mechanism for the zipper thing, inserting more CAGs and making it bigger, and all in a pill. That's kind of cool, okay? So they've done just phase 1, very early studies, but this is just showing that they can lower mutant Huntington. That's good. So they're going to a phase 2 trial first. They're starting in Australia at the beginning of the year. There has never been a treatment trial for Huntington's disease ever in history that has been open to patients in Australia. It is literally its own continent. So guess how those patients feel there? Yeah, they are excited. They're like ready to go. Come on, bring it, let's go. All the Australian sites, they've all got lists of patients ready, and they're ready, right? And they're like, why have you, like, yes, we have kangaroos and koalas, like, bring us HD studies too. So they're ready to go. They think if it goes as fast as we think, because they're ready to go in Australia, if they can nip this and zip this around, their sunny projection timeline is they may be able to ready to start a phase 3 trial in the US next fall. I think that's really optimistic, but they're gonna go for it. They're gonna go for it. Okay, that's it. So, sorry I went way over time, but I wanted to cover all these big announcements that we've had. You probably have seen these press releases and you're like, what the heck does this mean? I don't understand what this stuff is that we're talking about. I wanted to break those down for you. This is an unprecedented time in our history. It's exciting. Dr. Hernandez is going to continue that excitement. So sign up for a trial. We can only do this together. Thank you guys so much.
I wanted to hear it, so it was great. Thank you. Thank you, Dr. Sung. Great job.